Hey everyone, and welcome. So, for decades, trying to understand the human brain has meant making a really tough choice. And that choice usually involved animal testing. But what if all of that is about to change? Today, we are diving into a huge scientific breakthrough that could totally reshape how we do brain research and maybe, just maybe, bring an end to a really controversial practice for good. I mean, really think about this question. For so long, if you wanted to study something like Alzheimer's, you had to rely on animal models. But that comes with a whole host of problems, not just ethically, but scientifically too. So what if we could build a better, more accurate, and frankly more humane model of the human brain right there in the lab?
Okay, so to really get why this new development is such a game changer, we first need to get our heads around the two massive problems that have been holding back brain research. These aren't just small hurdles, they're major roadblocks that make finding cures so much harder. And here it is, the absolute core of the problem. We're using rodent brains to try and understand the human brain. But the genetic and physiological differences are just enormous. It means that what works in a mouse often doesn't work in a person, which is why they are such poor predictors. The result? So many failed clinical trials, tons of wasted money and time, and a much slower path to finding real cures. And this quote from Professor Nishadi perfectly nails the second big issue. See, even when scientists try to grow human brain cells in a dish, they have to use these special coatings to get the cells to stick and grow. And those coatings are made from animal proteins. The problem, as he points out, is that they're all different. It's like trying to bake a cake, but the recipe for your flour changes every single time. You can never trust that your results are consistent or reliable.
So, with those two big problems in mind, the animal models and the inconsistent coatings, let's turn to the solution. Because a team of researchers has developed something that tackles both of these issues head-on. It's really revolutionary. Okay, this is the headline. For the very first time, scientists have successfully grown active communicating brain cells, a neural network, on a platform that is completely synthetic. We're talking zero animal products, no animal brains, no animal proteins. This is a huge deal because it finally gives us a stable, consistent, and controllable environment for research. So let's break down what makes this new platform so special. First, and this is the big one, it is completely animal-free. That right there solves the ethical dilemma and the consistency problem. Second, it has what scientists call high biological fidelity, which is a fancy way of saying the cells grow and talk to each other just like they would in a real brain. And third, because it's so stable, you can run studies for a much longer time, which is absolutely critical for diseases that develop over years. All right, this all sounds a bit like science fiction, doesn't it? Building a brain in a dish. So how in the world do they actually do it? How do you get living brain cells to grow on something that's totally artificial? Well, let's get into the science of it. Okay, the star of the show here is a material called polyethylene glycol, or PEG for short. It's a super common polymer and it's chemically neutral. Now, for living cells, PEG is basically like trying to stand on perfectly smooth ice. There's just nothing to grab onto. So normally, cells just slide right off. They won't attach to it at all. The other key concept we need is a scaffold. And it's exactly what it sounds like. Think about it like the steel frame of a skyscraper. It provides the structure, right? It gives everything a place to go. A biological scaffold does the same thing for cells, giving them the support they need to grow into a complex 3D tissue instead of just a flat smear on a petri dish. And this is where the real magic happens. The researchers came up with this incredibly clever process. First, they mix that PEG with water and ethanol. Then they flow that liquid through these super tiny glass tubes. As it comes out, it hits a stream of water, which forces the mixture to separate into this intricate maze-like structure. And then, bam, a flash of light locks that porous structure into place. The result of that process is just wow. They've taken that slippery, useless for cells material, PEG, and turned it into a textured 3D matrix. It's the physical structure itself, all those little pores and channels, that gives the cells the handholds they need to grab on, to grow, and to build those functional networks. No animal proteins required. And as the lead author, Prince David Okoro, points out, the stability of this scaffold is the real game changer. It doesn't degrade or change over time, so they can run experiments for weeks or even months. Why is that a big deal? Well, it means scientists can finally watch brain cells fully mature, which gives them a much more accurate picture of how diseases like Alzheimer's actually progress in a real, functioning brain.
Okay, so we've covered the science, we know how it works. But what does all this really mean for the future? This is where we bring it all together and look at how this could usher in a new era of research, one that's more accurate, more personal, and, way more humane. Now, before we get too carried away, it's important to keep the current scale in mind. Right now, this scaffold is about 2 millimeters wide. So yeah, it's a tiny beginning, but the potential, well, the potential is massive. And the team is already working on ways to make it bigger. And this right here is the real-world payoff. Think about it. We could model traumatic brain injuries, strokes, Alzheimer's. And you could even take a patient's own cells, grow a mini model of their brain, and test drugs specifically for them. It's personalized medicine. All of this dramatically improves the reliability of drug testing, and it lines up perfectly with the push to finally phase out animal testing, directly helping to end animal suffering in labs. And believe it or not, the vision doesn't even stop with the brain. Professor Noshadi and his team are already using the same technique to grow liver tissue. He imagines a future where you can connect different lab-grown organs, see how a drug affects the whole system at once. It's a huge step toward understanding our bodies in a much more complete, integrated way.
So that leaves us with this incredible thought to chew on. This technology is a massive leap forward for more ethical and more effective science. But if we can model a brain and then a liver, what comes next? Are we looking at the beginning of a fully interconnected human on a chip? A system that could completely revolutionize how we develop new medicines? The possibilities are just staggering to think about.